today we'll cover those concepts, but I will make it easier. So if you have notes from last year, don't study them, because I made it easier than last year. Because last year I started with this lecture, so the students were fresh and alert. Now we're ending, so I have to make it easier and reduce the concepts. Okay, and because by now you should have studied many concepts in genetics, and also it's like putting the full picture together. This is the aim of this lecture. So we're going to talk about the regulation of gene expression. But to understand this, we have to remember first, what is gene expression?
RNA synthesis and
Transcription.
Okay, so this is basically gene expression is how you go from DNA to protein. And we said in all one when the gene, I want to say gene and DNA, but gene. When the gene expresses itself, it means it is going from just being a DNA sequence to being what? Protein. Okay? So we studied this in Module 1. Here we're going to talk about the regulation of the gene expression. How we can make the gene expression happen. So how we switch it on, how we switch it off. How we can make it go faster and how we can make it go slower. Right? Yeah, move your phone, please. So this is what we will talk about today. And this regulation, okay, so this is a journey going from this tightly compact chromatin to the protein that can do a function, right? This is the journey of gene expression. How we can regulate this process at multiple stages, okay? And today we are going to talk about 3 stages. We will regulate the transcription, so make the transcription happen faster or slower, or we can switch it on, switch it off. Also, we can regulate the lifespan of the messenger RNA, make it live longer, okay, or shorter, because, you know, the lifetime of this messenger RNA will determine how much protein I'm going to make. The longer it will stay, the more protein I will make, right? So this is another level. The third level, I can regulate translation. So I can make translation happen, or I can block it. So all of these factors will regulate the making of the protein. Okay? And this is very important to understand, because this journey will change the DNA material into a protein that can have a function. Not only that, the making of this protein will make our cells different. What is the difference between the different cells? If we look at cells, if you take a cell from, let's say, the liver, and a cell from the, what? Give me another organ. Pancreas, or the kidney. Okay? What makes these cells different? The DNA? So do they have the same DNA?
Yes.
Do all cells in our body have the same DNA? Yes. So all the cells, they have exactly the same genome. Okay? But despite that, they are different. So what makes them different?
Function.
The gene expression. Because different cells will express different protein. Even though they have the DNA, they have the genetic material to make all the protein, they don't do that. They will express only the proteins that will make them differ. Okay? So, this is what we call differential gene expression. And what do I mean by differential gene expression? It means that different cells will express different gene, even though that they have the same genome. Clear?
Yes.
Okay. So how do we regulate that? We can regulate this at many stages, and we will see later as we have outlined here. So now I'm going to talk about the stages of regulation, and we will start with the first stage, the regulation of the chromatin. We have learned from, let me show you this. We have learned from Mole 1, and no, the tightly packed chromatin will not be active in gene expression. Why?
Because the helicase can't access the nucleotide.
Yeah, because the enzyme will not access, we cannot read the sequence, how we are going to transcribe it. So the first thing to express the gene, you need to do what? Unpack it. So you need to loosen the chromatin structure. How can you make this chromatin structure loose?
Because
No, this is at the second level. This is no, this is something new that we did not study before. Okay? So this is before you go to the DNA level. Here I'm talking about the chromatin level. Okay? Helicase will do its job with the double-strand DNA. Once you have a loose chromatin with the double-strand DNA, you have to separate the two strands with the helicase. What you said is correct, but this is when you're already doing transcription. But before doing transcription, you have something like this. You cannot read it, you cannot transcribe it, you cannot do anything with it. So what you need to do is make it like this. So you need to go from the heterochromatin to the euchromatin. Right? This is what we learned in Mol 1, remember? So how we can go from the heterochromatin to the euchromatin?
Acetylation.
Yes. So we need to do some chemical modification to the histone and to the DNA. Okay? There are many types of chemical modification. At your level, you should know two things. You should know that acetylation, those green things are acetyl group, which can be added to the lysine, amino acid lysine, which is present in the histone. Adding this acetyl group to the lysine of the histone will make the chromatin loose. It will unpack the chromatin, which is needed for transcription to occur. Okay? So acetylation of the histones will activate transcription. It's easy to remember that acetylation activate. Okay? So this is the first modification of the chromatin by adding acetyl group to the histones. What are the histones? We learned from Mol 1. These are the histones. They are the protein around which the DNA is tightly packed. Are we in a kindergarten?
ما بعرف طالبة.
So what are these histones? We have 8 histones. As you can see, they form what we call octamer disk. Let us zoom into their structure before we continue with the modification. It's a question that they always like to ask in the USMLE. So you have an octamer, so you have 8. They form like a disk of histones. We call them the core histones. This core histone is made of 4 types of histone, which are H2A, H2B, H3, and H4. Okay? So this is the core of the histone. But also we have a linker histone, which is H1, which is outside. What is the function of this linker histone? It's make the packing even harder. So it make the packing stronger. Okay, so this is the linker histone. It will facilitate the formation of more compact structure. Okay, so this is concerning the structure, the basic information that you should know about the structure, the octamer and the linker, and you should know the name of each histone. The modification that we are concerned with today, one type is adding acetyl group. So acetylation will make the chromatin unwind. Okay, so we're going from the hetero to the euchromatin. Therefore, we are facilitating the transcription. The second type of modification is methylation, but this time it's the DNA methylation. So we're not talking here about histone. We are talking about the DNA itself. So if we add methyl group to the DNA, and specifically it is added to the C and the G, so it's the C and the G bases of the DNA. Once they are methylated, it's going to activate or inactivate the transcription? Inactivate. So you see the lamp here, it's off. It means that the transcription is switched off. So methylation, if you want to remember an acronym, methylation will mute the gene expression. So methylation will mute. So in order to activate the transcription, what you need to do with the methyl group, you need to remove it. Okay, so you need to do demethylation. So that's why it's written here that acetylation and demethylation, these two chemical reactions, okay, but you should know that acetylation of histone and demethylation of the C and G DNA nucleotide, these are going to convert the chromatin into less tightly coiled structure. Therefore, it's going to favor the binding of transcription factors, which is the second part of the story for transcription to occur, and therefore it's going to activate gene expression. Okay? Because why we need to have the structure loose? Not only to have the access for the enzyme, but also to have access for the transcription factors. Okay? Have you learned about transcription factors in genetics? You should have. And I think I mentioned them in Mole 1. But anyway, we'll come back, we'll talk about them in detail today. What are these transcription factors? They are proteins that bind to DNA, to certain region of the DNA, and they usually turn transcription on, or they can turn it off. Okay, so these are the transcription factors. Okay, I will come back to this at the end, but let me just quickly show you here. So in every DNA, you have the coding region, which is, we have the exon, and then we have the intervening intron, the non-coding. Here we have the promoter, right? And then we have these control elements. Okay? We have these control elements. What are they? They are the ones that are going to control transcription. How they will control transcription? I'll show you another photo. This one. Here. Okay. So this is my DNA. You can see sometimes you have a folding of the DNA on itself, or because we are trying to bring two regions together. But what's happening here? And these transcription factors in brown here and other these transcription factors will bind to the RNA polymerase, and they will initiate transcription. Okay, so they bind to the DNA and to the polymerase, or they interact, if you want, with the polymerase, and they bind to the DNA and will initiate the transcription. Okay, so let me repeat. For transcription to occur, you need to unpack the chromatin in order for it to be accessible by the transcription factors. The transcription factors, they are protein that will bind to the control element, and they will interact with the polymerase, and they will initiate or regulate the transcription. Is it clear? Okay, I will come back to this in more detail, but now let me finish. So we talked about the modification, and one thing that you should know also about this modification of the chromatin, so we call them as in general chromatin modification. These chromatin modification can be passed from one generation of cells to another. They actually can be inherited. Okay, before we always talk about genetic factors. Now there is epigenetic factor. What is epigenetic factor? It is, one example is this chromatin modification. When we modify the chromatin, we are modifying the gene expression. Therefore, we are modifying the level of proteins we are making. Okay? These epigenetic can be passed, can be inherited, and then they can control, the future generation of cells, and they can be passed from parents to children. So we call this mechanism epigenetic inheritance. Why epigenetic, not genetic? Because it's not affecting the actual DNA sequence. It is affecting the structure of the chromatin. Okay? So epi means above, so it's above the gene. It's the control of the structure of the chromatin, not the sequence of the DNA. Okay, so these factors will control the transcription initiation, so the start of the transcription, because they will make the DNA less or more accessible by the transcription factors needed to transcription to occur. We mentioned the control elements. So what are the control elements? They are part of the DNA. Okay, so they are segment of DNA. Is it coding or non-coding? Non-coding. What do? They serve as binding site for what? Transcription factor. To do what? To regulate transcription. Okay? So it is the presence of these control elements and their binding to the transcription factor that will do the precise regulation of gene expression in the different cell types. Okay? So why a cell, we come back to our question, why a cell from the kidney is different from a cell in the liver? Because they have different transcription factors. Okay? The presence of these different transcription factors, and then they interact with the same gene, okay? Then they will lead to different proteins being expressed in the different cells. So there is a nice picture, but I removed it from this slide. I wish I did not. They show you that it is actually the different transcription factors that make our cell differ. Yes.
If the control elements are part of the DNA, it means they also the same control elements, because you mentioned only transcription factors are different.
Yes, they are the same, but they differ in the different gene. But the different cells will have the same control elements. Yes, because we basically have the same DNA in every cell. But these control elements, they differ for the different gene. Okay? Type. So we have two types of transcription factors. I'm not going to go into details, but we have the general transcription factors, which are needed for what we call basic transcription. And these are the ones that are needed for every gene to be expressed. So for every gene to be expressed into the protein, we need to have those general transcription factors, which are common among all cells. But also we have specific transcription factors. And these specific transcription factors, they are specific for different genes, and also they are the one not to initiate the transcription, they are the one that do the fine-tuning. So they make the reaction, it's not the action, they make the transcription go faster or slower. Okay? So this is, if you hear or listen about the general and the specific transcription factor, this is a difference. So general is common for all genes, and the specific is only for specific genes. So I mentioned the proximal. So what is the difference in English between proximal and distal? So proximal, it is near to what? Near to the transcription site, the start of the transcription, okay? Or the promoter if you want. The distal will be far. Okay, how far? Look at it here. What do they mean here? It's not that close. It's very far. It can be up to 2,000 base pair away. Not only that, this is another question that they like in the USMLE. And no, these distal control elements are called also enhancers. And these enhancers, they are not only upstream, they can be also downstream, they can be in the introns. So they can be placed anywhere, and still they will control the transcription. And this is why I showed you in a previous picture. Is it we have this bending occurring. Why we have this bending occurring? Because the enhancers are very far. And for transcription to start, you need to have everything in one place. So the DNA has to bend in order to bring this far sequence, to bring them near to the transcription site. Okay? So this is why we have the bending, to bring the distal control element nearer to the transcription site by bending the DNA. Right. What are the transcription factors? Again, if you want to say, we talked about the control element. We said we have distal and proximal. What about the transcription factors? We said there are general and specific. Okay, among the specific, also we have something called activator. Sometimes we like to call them activator. What's an activator? It's a transcription factor. Okay. It will bind to the enhancer, the other name for distal control element, and stimulate the transcription. Okay, so this is an illustration of this one here in red and purple is a transcription factor. Okay? So the transcription factor usually have two domains or two parts, if you want to call them. One part will bind to the DNA or to the control element, if you want, of the DNA, and the other part will do the work, which is initiating or activating transcription. Okay? So that's why we say they have two domains, DNA binding domain, so a part of the protein of the transcription factor will bind to the control element to make sure that, you know, this is the right gene. And this one is the one that's going to do the work of activating the transcription. Clear? Okay. A good example is what we have studied about gluconeogenesis. Okay, what is gluconeogenesis, ya Bashir?
Making new glucose from the liver.
Okay, synthesis of glucose from non-carbohydrate precursor, right? So if you are fasting in Ramadan, let's say, your blood sugar will go down, so gluconeogenesis will be activated. In biochemistry, we have learned if you want to activate any pathway, what would you do to activate the pathway? You will, yes, you will activate which enzyme? In any pathway. In any pathway, if you want to activate this pathway, which enzyme would you activate? Which enzyme? Of which step? Rate-limiting enzyme. Okay, so you need to activate the rate-limiting enzyme. Also in mode 1, we have learned that there are two things or two ways to activate the rate-limiting enzyme. One fast, one slow. Right? What is the fast way? Yes, so you make the enzyme more or less active. And what is the other way? Making more. Making more of the enzyme. So here we are going to talk about making more because we are talking about gene expression. So you're fasting, your blood sugar goes down, the body is trying to maintain the blood sugar, so your pancreas will secrete glucagon, and glucagon want to activate gluconeogenesis. To do that, it will activate the rate-limiting enzyme, which is Pepsi kinase, right? Phosphoenol pyruvate carboxykinase. How we are going to make more of this enzyme? Glucagon will bind to its receptor, which is a membrane-bound receptor, okay? And it's going to activate cyclic AMP. Cyclic AMP will activate protein kinase A, and نحن ما كملناها هيدي بالمول 1. It's going to activate a certain protein called CREP. What does CREP stand for? It is for cyclic AMP response element binding protein. From the name. So it's a protein that is going to bind to a control element. So what does this mean? What type of protein? Excellent, Saqfulo. Transcription factor. So to make more of this enzyme, we need to activate the transcription factor that will bind to the control element of the enzyme gene in order to activate the transcription of the enzyme, to make more of the enzyme, to make more glucose. Okay? So this is one way. Yes? The other way is not when your blood sugar goes down, when you're under stress. If you are sitting an exam or yes, so you are stressed out, especially long-term stress. Which hormone is secreted by your body? Long-term stress is cortisol, right? So cortisol is a glucocorticoid. It will, we learned this in Mol 2 also, that cortisol can upregulate gluconeogenesis by activating the rate-limiting step. But how does it do that? At the genetic level. I think it was written in the slide at the genetic level, so cortisol will bind to its receptor, but this time the receptor is intracellular. And this complex, اللي هو الcortisol plus the receptor, will go inside the nucleus. And now where is the transcription factor? هو هيدا الcortisol with its receptor, okay? اللي هو called GRE receptor, because it's going to interact with the GRE control elements. Okay, so we have glucocorticoid response element. Okay, it will bind to cortisol and it is receptor. Not this one. Okay, so I repeat. So cortisol will go inside the liver. It will bind to its receptor. Then the complex will enter the nucleus, bind to the GRE because it is the protein. That is going to act, or this is the transcription factor that will bind to the GRE response element, okay, that will activate the gene, okay? So, this is the transcription factor. What is the transcription factor made of? Cortisol plus the receptor, okay? And what is the name of the control element? GRE, okay? So it is glucocorticoid response element. So we have GRE and we have, what's the other one? CRE, okay? Sorry? Zinc finger, I didn't want to go into detail. Remember when I talked about the domain, we said the protein or the transcription factor has two parts. One part that will bind to the DNA and one part that will regulate transcription. This part usually has a specific shape, and there are classes of different shapes. The zinc finger, so it looks like a finger and it has a zinc in it. Okay, it's a name they give it to describe the shape of this domain here, which is the one that binds to the DNA. Okay? We have also leucine zipper, because it looks like a zipper and contains leucine. So we have different types of domains, okay, which are the parts that are going to bind to the DNA control elements, okay, part of the transcription factors, okay? So in other words, don't worry about it. So this is the first part, which is how to regulate transcription. What about the other parts? How we can regulate gene expression? We can regulate it by regulating the lifespan. Okay, so it's written here. So we can the 3' untranslated region of the messenger RNA will determine how long the messenger RNA will live. Also, we can regulate the alternative splicing, and this is why we have one form of the protein expressed in one tissue but not in the other, because of alternative splicing. If you recall from mole 1, right? You remember from mole 1, alternative splicing. This has to do with which part of the gene is recognized as intron. Okay? So this is another way. So we can regulate post-transcriptionally or after transcription, we can regulate the lifespan and we can do alternative splicing. What about the third level? Translation. We can regulate it at the level of translation. How we can regulate it at the level of translation? We have two ways. This is one way, and it is giving you the example of ferritin. What is ferritin? It's the protein that stores iron. Iron is toxic. Okay, if we have excess iron in the body, we will store it inside ferritin. So we don't need ferritin unless we have iron. But once we have iron, we need ferritin quickly. So how we can make ferritin quickly? By regulating the translation, not the transcription, because we cannot afford for the iron to stay in the blood. We want to store it. So this is why the regulation of the Gene expression for ferritin is at the level of translation. So the messenger RNA is made and it's kept there, but it's switched off. Why it is switched off? Because we have this protein that is blocking it. It's like a key locking the translation. How can we remove the key? Iron. So it makes sense. Once iron is there, it means I need ferritin. So iron will be the key that will unlock the door. It will remove the lock here. And once we remove the lock, what will happen to translation? Translation will occur and we start making ferritin. So once we have iron, we will inactivate that that binding protein which was locking. It was active because there was no iron. It was binding to the messenger RNA and blocking its translation. When iron is secreted, it will bind to that purple thing, which is the binding protein. It will bind to it and inactivate it, and therefore translation now will happen because the lock has been removed. Clear? So this is one way of regulating translation, okay, by using the binding proteins which can lock the translation. Yes?
Doctor, is IRE a control element or are they only in the transcription?
IRE is the control element for the messenger RNA, okay? So it is it's a response element, iron response element, okay? And here we have the iron response element binding protein. The purple thing is the binding protein, the protein that will bind to the IRE, okay? Which is the iron response element. Clear?
Okay, but do we consider it a control element like in transcription or we do not?
No, because here we are not regulating transcription, we are regulating translation. Okay? So although it is a control element, the same word is used, but it is a control element for translation, not for transcription. Okay? Yes. Yes.
Doesn't having intracellular iron mean that we already have iron, we don't need to make more? Like, why does iron activate intracellular?
No, because we need to have some stores of iron. Always we should store some iron in the body. Okay? The way our body works, and we have iron in the hemoglobin, but we should also have some iron stores. You'll study this in your heme block next. Yes.
You said two slides
Yes. Lifespan, yes. Yes. It will have, it's the UTR, antranslated fusion, will dictate a long life. Yes, will have a longer life than other proteins. Okay? Yeah. The secret time. Okay, the last part is also about regulating translation, and this is a new field of research. We talked about RNA in Mol 1, but we didn't mention all type of RNA. There is short non-coding RNA also, which are called microRNA, and we have also the small nuclear. Yes, so we have microMe, and we have the small interfering RNA. So we have Me and cRNA. What are they? They are short sequence of RNA, also non-coding, but they have, what they can do is they can interfere. Interfere with what? With translation. So this is the mRNA, the microRNA. It can bind to the messenger RNA, mRNA, and it can either degrade it or it can block the translation. So this is what we call RNA interference. And this is a very new area of research. They use it in the lab, so we can make in the lab small small interfering RNA. The method is called RNA interference. They can use it in the lab, and they're trying to edit the genes in it. So I'm not sure if you heard about the new technique called CRISPR. So in CRISPR, they use the cRNA. So they have a short RNA that interferes with translation. So by blocking or degrading the RNA, then you don't translate it, then you don't make the protein. So they are using it as a method. They are using it as a method of gene editing to treat people with disease. They're still at the early stage, but there are some promising results coming for diseases such as Huntington disease. Okay, I'm sure you heard it in the news about use of CRISPR technique to treat Huntington disease. So this is employing the control of translation by using RNA interference. Okay? With this, I conclude.